XFELs. So Jimmy, if you are ready, then please share your screen. Okay, perfect. Yes, so we are curious to hear more, to learn about warm dense matter, and the stage is yours.
Thank you. Good evening, everyone. So today I'll be speaking about a review article, which is Warm Dense Matter Studies with X-ray Free Electron Lasers. And myself, I'm Jimmy Guruprasad. I'm a PhD research scholar at Optomechatronics Laboratory at IIT Madras. And this is actually a very different area which I have been covered. So just correct me if I'm wrong. So, warm dense matter is nothing but it is a state of matter which is like very strongly coupled, and it is having a moderate or to high temperatures ranging from zero point 0.1 to 100 electron volts. And to get a better idea, it is like an intermediate state between the condensed matter and the ideal plasma, where in the condensed matter it usually assumes a temperature approximately a very low temperature, while in the classical plasma physics there is a weak coupling, but in warm dense matter there is a high correlation between the ions, so we cannot consider it as an ideal plasma. So probing these kind of dense matter is very difficult in a laboratory scale, but it is being possible nowadays. And if we look at the warm dense matter, it is like sitting somewhere around the Earth's core, and but not to the sun's core. And this and another thing is that it is having an intermediate temperature, as I said, which is like 1 to 100 electron volts, and it's having high density, and it is partially degenerate and strongly coupled. When we tell about the warm dense matter, we usually it is naturally occurring inside the large planets like Jupiter, and you can see this is very high dense, but this is the warm dense region, and it occurs in white dwarfs, brown dwarfs, and it the interior what and if you look at the pressure which it is generated, it is approximately like very high pressure, this warm dense matter 16. So these, I suppose that high heating and dynamic compression and implosions, these can be achieved within the laboratory scale, which is marked in the red. And Coming to the XFEL parameters, as we all know that there are different free electron lasers in the world. And if you look at most of the repetition rates of the free electron lasers, it is very less, like not into the megahertz range, but it is in the hertz range. So we when we look at that, it is a limitation, because one thing is that we need hard X-rays and to probe these kind of matters for deep penetration into the density of the material matter, and we need a high repetition rate. So if you look at the different free electron lasers, the European XFEL and LCLS is having uh, hard XFELs, X-rays, basically. And today I will be focusing on these two XFELs. Uh, so when we compare it to the synchrotron, it is having like approximately uh, a million times higher brilliance compared these XFELs are having. And this is an advantage for probing these warm dense matters. So we know that at European XFEL, we learned that there are three self-amplified spontaneous emission uh, sites. So we have the high energy density science, which deals with the warm dense matter. So these are the different other capabilities of the other. So you can see that the high energy density is being probed, and we can see the ultra fast processes and extreme states. So basically, the first one is focused on biological structures and femtosecond chemistry and physics, and two is more focused on high energy when extreme density physics, which is for the warm density matters, and the third one is for soft X-ray spectroscopy. And you can see that these are using the hard X-ray and these two are using the soft X-rays. So in the high energy density at the laboratory scale, we are able to achieve it through by laser compression. And we have the diamond anvil cells for that. And we can heat the samples isoporically so that we can reach a solid density and a temperature up to kilo electron volts. So this in the European synchrotron, we are having like basically two different lasers, that is the relax and the dipole laser, which is able to achieve this kind of compression for high density for and warm density studies. So as I said, this is the diamond analysis where which are basically used for studying the inertial confinement fusion, and dipole lasers are used for heating the samples. And coming to the parameters, we can see that this is a long pulse laser, and this is short pulse, and it is and the temporal regime is given. And this actually this is the interaction chambers. We have two interaction chambers for doing the experiments at the synchrotron. Sorry, the XPL. So how to create this warm dense matter? So one is by the heating with the free electron laser beam. One is through, as I said, those two lasers which are having the short pulse and the long pulse. So this is the timeline that it had been installed throughout the years. So the first warm density matter experiments were done at Flash, and progressively it went through the two-color demonstration and the X-ray heated diamond anvil cells at European XRPL. So if you look at how it is being done, for if we are heat pumping with the XFEL beam, so it will isotonically heat these samples up to like 100 electron volts, and this photoelectrons and Auger electrons will generate ionize more ions, and via collision it leads to high temperature and densities. If we look at the long pulse lasers, we'll able to generate this dynamic compression waves, which are like shock or ramp compression waves, and we are able to achieve a high pressure similar to the astronomical objects, etc. So then the femtosecond to picosecond lasers, which are used for energy over 1 joule, and it will heat the thin samples to the warm dense matter conditions. So the warm dense matter can be probed at the free X-ray laser electron lasers by using different methods. Like we have the probe and the pump and the probe methods, and we have like the small angle imaging and wide angle, and so if you look at the different capabilities that we have, is like at a different scales we can probe this. For example, with short pulse we can probe these kind of effects like electron-electron equilibrium, ion-ion equilibrium. So for different applications, we have to go with a different kind of probing methods. So basically, these are the probing methods which is given in this review, which is the phase contrast imaging and small angle X-ray scattering, then X-ray diffraction, then X-ray absorption near edge structure, then extended X-ray absorption fine structure, X-ray Thomson's scattering, then resonant inelastic X-ray scattering, X-ray Raman scattering, X-ray emission spectroscopy. So we coming down the scale, we can probe smaller atoms, and basically these are the key. Then coming to the planetary and stellar physics. So this is an active research area where the warm dense carbon is studied. So basically, these icy giant planets and all are having like a diamond precipitation and magnetic field structure due to the liquid metallic carbon. So carbon is very much abundant in these kind of planets, and it is also known to exist in the cores of uranium and Neptune. So studying this warm dense carbon is highly enlightening. So basically, one study which has been conducted as a European free electron laser is through the liquid structure of carbon, which was probed with this 18 kilo electron volts. As you can see that we are having up to 25 kilo electron volt, and 25 femtosecond was used for that, and up to 150 we can go. So like 20 micrometer spot size was used. And the dipole laser, which I told, like which had the capability for in the nanosecond lane, but it's a long pulse duration. And what we were able to see is that in the ambient condition, we are having like a broad amorphous glassy carbon. As we increase the temperature, sorry, increase the pressure, we can see that around this 76 GPa to 105, 106 GPa, we are having this diamond formation as we begin to compress. And in after lots of compression, it becomes liquid state. So this was demonstrated, and another study was based on the X-ray absorption near KH. So what they did was like they took a aluminium sample which is very thin, like 100 nanometer, and they passed one. This is with a short pulse, like 120 femtosecond, and at 800 nanometer and 86 joule per centimeter cube. So as you can see that first it is crystalline. We can see the KH here, but as we increase the can see that it has becoming from solid, it is gradually going to a vapor state. So 1s to 3p transition is being shown here. Then another application for why it is very important to study this warm dense matter is for inertial confinement fusion plasmas. So here the deuterium and tritium are like they are being done, reaction fusion is being done, and a neutron and energy is released. So how it is done is in like different forms we can do it, like in diamond anvil cells that I said, as well as there is a holorium kind of thing. So like what we are basically doing is like with lasers or X-rays, we are symmetrically heating this, and we are making the fusion, and it is being ignited. So the the thing is that for studying this kind of it is very much important, because sometimes this laser plasma interaction and this instabilities come a lot. So studying this is very important, and it is being we are able to probe these kind of things in the European experience. So as I said, there are different phases for this. There is an initial compression, then there is an isotropic compression, And there are different ignition, different types of ignition, shock ignition. And as I said, this is one of the research areas where the laser plasma instabilities are studied. And these are the initial confinement facilities around the world where the studies are going on, prominently this NIF. So also it is also very useful in studying how the life originated. We all know that once a meteor struck the earth and the old life forms like dinosaurs disappeared. And but how did we evolve? Like when such kind of pressure is generated and there are like different pathways any molecules can take, like the organic molecules like carbon, nitrogen, oxygen, and these kind can form amino acids, nucleobases, fatty acids. So this kind of research is also very helpful in determining how we all originated. And the main thing is that compared to synchrotron, we are able to see the atomic resolution very better. And before the synchrotrons and X-ray electron lasers started, only a few proteins were known. But now we are able to also study different structural biology with these techniques. And now coming to what is the future of it. So we are in this current stage. We are using the femtosecond, nanosecond and all. But now the idea is to go to the attosecond scale. And there are major upgrades also happening. Uh, so and also there is one disadvantage that present we are having only three, but synchrotron has different points. So that is also like a signal increase in the capabilities. So thank you.
Yeah, okay. Thanks, Jimmy, for this nice overview presentation. Are there any questions? I mean, Jimmy touched on various different topics, so you might have a question to one of these different aspects. And then, okay, let me ask one first question. In the beginning of your presentation, you mentioned high repetition rates, that would be important. And in this table, you showed that most FELs had repetition rates in maybe in the range of between 50 and 120 hertz or so. Only XFEL now has higher repetition rates, but it comes in a strange burst scheme, and also actually LCLS2, the next generation LCLS, has the goal to produce like continuous 1 megahertz repetition rate pulses. And they are not there yet because this is very challenging, but it's getting there. But my question is, why do you think is it really important to have high repetition rates for these kind of studies?
I am thinking that it is for probing the transient state very effectively.
You mean in a pump probe scheme where
Yes,
a
yes,
first pulse excites and the second pulse probes it?
yes.
Yeah, I mean, there the repetition rate is not so important. There it's more important that the pulses are short enough such that like the first pump pulse triggers some dynamics in a with sufficient short time resolution, and then such that the second pulse has the chance to probe whatever short dynamics is happening on a time. Yes, exactly, on this. Possibly, if it's electron-electron correlation effects, then ideally we would want pump probe delays between the two pulses on the order of femtoseconds or even below. And I mean, we have heard a first presentation about all attosecond pump probe measurements in water. And on Wednesday, there will be another presentation on attosecond. So this is indeed interesting, but this has nothing to do with repetition rate, right? This is just the time difference between the first pump pulse and the second probe pulse. But that time delay can be much shorter than the time it takes for the next pair of pulses to arrive. Yeah. I mean, even if your repetition rate is only, let's say, 10 hertz, so every 100 milliseconds comes a laser pulse, then still you can have a second laser pulse that's timed to be just a few femtoseconds after the first pulse in a pump probe scheme. So, yeah, my understanding is that repetition rate is actually not so important for these studies because, because I mean, we're talking about very high intensity in the pulse. So these high intensity pulses likely completely destroy your sample. I don't know if have you read about this? Like,
Yes.
so it needs some time to replace the sample by moving some thin film by a few millimeters into the focus, or I don't know, sometimes people use liquid jets or something, but the repetition rate can actually not be too high, otherwise the next pulse would hit the already damaged sample and you would start interacting with the damaged sample. So it actually needs some time to like replenish your sample. Yeah, okay,
But
that's
maybe like more hard X-rays are available. That is why maybe when because like the other synchrotrons are having like not in the repetition rate, but use of availability of hard X-rays to probe.
okay, sure. And why do you think hard X-rays are particularly advantageous?
Because it will be able to penetrate denser media matter than the soft X-rays.
Okay, yeah, yes, that you're completely right. Sure, yes. So yeah, this is actually another question that came to me, like when you mentioned this research, the ongoing research in the context of nuclear fusion plasmas. I mean, I like the most prominent facility is probably the National Ignition Facility in the United States. But this is not free electron lasers, I think. This is some nanosecond high power infrared or visible lasers. So the question would be, how exactly do XFELs help for studying fusion plasmas?
Basically, for the equation of states, they were mentioning in the review paper, like it is very difficult to model the equations. So so they are doing it in these kind of facilities where they generate these shocks and then probe through this X-ray Thomson scattering, etc. So the different density of states, etc. I'm
Okay,
not
so
very sure
I see.
about it.
So the XFELs are used more for diagnostics, right? Not
Yes.
really to trigger the plasma or fusion reaction, but it's more for diagnostics of what's happening in the plasma, right?
Yes. And there
Yeah.
is like implosion, like when these deuterium and tritium, when they are sitting together, and when the implosion is happening in a spherical way, that can be very challenging. But it should be in a equal, compressed equally in the all directions on the sphere, not on so that laser plasma instabilities and all they can monitor. Is what how we can change the laser parameters so that spherical implosion happens. As you see here, this should be spherically, it should compress equally in all directions. But sometimes it is that two or three directions having more compression, so it will not be self-sustaining, the fusion. So that kind of instabilities, they are still being studied actually.
Okay. Okay, yes. Maybe one last question from my part. Like also in the end of your talk, you mentioned that like one of the new directions is going towards even shorter pulses, attosecond pulses. What's exactly the advantage, or what's the goal of using attosecond pulses for this type of warm dense matter research?
So it will be for not for the warm dense matter, maybe for the general studies, how it is. I just mentioned that because like about the nature updates, like how it can probe more the as I said, like for the dynamic structure of the biology and all. So more if we are going with a better higher resolution, so this attosecond may be like very helpful for the nuclear process, maybe like, as I said, the fusion reactions.
Okay, for actually like resolving the dynamics of the nuclear reaction, of the nuclear fusion reaction. Is that, yeah, okay. That's, of course, yes. That is likely to happen on a very fast time scale. Yeah, because, I mean, yeah, that's a good point. I mean, usually people always motivate attosecond pulses by wanting to resolve electron dynamics, because electrons move very fast, right? So if you want to, for example, see how the electron cloud moves in a chemical reaction, I mean, this is femtoseconds or even faster. But of course, as soon as nuclear reactions can occur, since these are so energetic, then even the motion of the nuclei becomes extremely fast because of these extremely strong forces that are acting in nuclear processes. So then actually even then attosecond resolution might be useful even to study the nuclear motion. That's yeah, this is usually we don't think about that, but of course nuclear physics also exists and and triggers much faster and much more energetic processes. Are there any more questions? Okay, I mean, if this is not the case, then thanks, Jimmy, again. Very nice overview presentation. It went a bit fast, but I think still very interesting.
Thank you.
Let's go to the next presentation, which will be given by Jayesh. So please, Jayesh, share your screen. Jayesh, yes. Presentation is gone again.
Hello, am I audible?
Yes.
Can you see my screen?
Yeah, but not presentation mode yet. Yes, now we see it. Okay, so please go ahead.
Good evening, everyone. I'll be talking about ultrafast relaxation of photoexcited superfluid helium nanodroplets along with Dr. Dev Prasad. So this paper was actually written, and then hopefully we'll give a brief understanding of what we felt we learned from this paper, and then hopefully can have a discussion about it. So the content of my talk will be, first we are going to see what system is under study and why we are studying this. Then we'll go into the experimental method. This experiment was done in Fermi FEL at Electra, and the process involved is the HGHG, which is the high gain harmonic generation, which I will give a brief overview of. Then we'll be looking at the results of this experiment, which will be discussed by Dr. Dev Prasad, and then look at this conclusion. So, why doing this experiment is basically in this experiment, what the authors have done is they have just used superfluid helium nanodroplets, and it was excited by femtosecond, and then probed again by another laser to study its relaxation dynamics, the time scale in which the relaxation occurs, and some applications of this process. Studying why we want to study this thing, some applications would be including studies about the atmospheric sciences, the radiation damage, the light, the light harvesting, and the photocatalysis process. Now, why helium nanodroplets? What I understood that the author they use very common, one of the common things used for their experimental work is helium nanodroplets, because it is one of the most interesting experimental matrix and inert gas. The helium atoms usually have very simple electronic structure, just 2 electrons in it, and then they have very weak interatomic binding, and usually they are size independent and a homogeneous structure, and it's a very ideal model if you want to probe different photodynamic studies. Even though they are superfluid, that is, they don't usually lose kinetic energy when they flow, and these experiments usually occur at around very low temperatures, order of 2 Kelvin. So the experiment method used in this paper was a time result photoelectron spectroscopy. So in this case, we have a pump XV pulse from the FEL, which excites the pulses, which excites the droplets, and then use a ultraviolet probe, from which you generate electrons, and the energy of the electrons is that basically photoelectron spectroscopy is used to detect the energy of the electrons and study what electrons are emitted from this process. And then simultaneously, along with this experiment, a theoretical work which using time-dependent DFT method was also done, and then they are compared with to see whether the theory goes along with the experiment or not. So as I mentioned, in the experiment, the source was the FEL from Fermi at Electra facility. So just to give a brief overview, in the FEL, usually you have a modulator, which is a very short undulator, short length undulator, in which you do have electrons from a linear acceleration flown into it, and also which is coupled to a seed pulse laser. As uh, as in contrast with the SAS method, where itself the electrons form self bunching. Here, the seed pulse is the one which is interacting, a seed pulse from external laser, which is interacting with the electrons in the modulator. The only necessary condition for it is that the wavelength of the seed must follow this undulator resonance condition. So you have if this condition is followed, you have modulation of the electron beam energy, and once it passes to a dispersive section, which is actually a magnetic chicane, this electron beam modulation is converted into a density modulation, which forms short micro bunching of the electrons, and then the short bunches of the electrons are moved across this radiator, which is a longer undulator length, where you have the amplification happening, which releases a FEL pulse. In this case, the advantage is that this FEL pulse can be of the order of, can be a harmonic of the seed pulse. Hence, we have a harmonic generation, and the gain is very high, and it's called a HG. And so the important advantage of is that the FEL pulse is usually a very, very short wavelength. Basically the bandwidth is quite small, and it's dependent of the input seed laser. So for this particular experiment, these are the parameters which are used, where you have a linearly polarized XV pulse, which is set to fifth harmonic of the seed laser with a pulse width of 100 femtosecond and a very short bandwidth. And you have a pulse energy of around 6 microjoules, while the probe, which is a frequency triple directly from the seed laser, which is around 775 nanometers, ultraviolet, and the energy of this pulse comes around like 4.8 GeV. I'll explain the significance later. And usually you have around a pulse width of 170 femtosecond with a 7 microjoule pulse energy. So this is how the experiment, just to give illustration of the pump probe spectroscopy. So yeah. So here you see in the gray area, that's the absorption spectrum of the helium nanodroplet, which shows a broad absorption roughly around 21.6 eV. So usually you have a pump, which is shown by this red arrow, which excites the system to this energy. But there exists some sort of an internal relaxation method or internal conversion method, which leads the state to be relaxed to this 1s 2s state, from where you have a probe pulse around 4.8 eV, which excites it beyond the ionization potential of the helium nanodroplet, which is around 24.6 eV. And whatever balance energy is there, which comes out in terms of the electron kinetic energy. So if you look at this internal conversion relaxation, which will be explained in more detail by Dr. David Prasad in the coming slide, what we understood is that if theoretically, if you look at it, between these energies, you do have the if you plot the potential energy versus the intraatomic distance of the different states available there. If you use a pump to excite it roughly around 21.6 eV, which comes into the 1s 2p state, due to this mixing of bands or crossing of the bands between these this green B1 state, the 2pxy state, and the red, which is the C state, there is this internal relaxation of the electrons directly into this state that also takes some energy along with it. And then you have the probe, which actually excites it from this 1s 2s directly into the continuum. So about the results of this thing and in detail explanation, which will be done by Dr. David Prasad.
Thank you, Joyce. Can you go to next slide? Yeah. Good evening, everyone. Good evening, Professor Masai. Thank you, Joyce. It was a very nice explanation. Am I audible, right? It is clear?
Yes.
Yeah. Okay. So now I'm going to continue further with this ultra-fast relaxation of helium nanodroplet experiment. So the time result photoelectron spectra measured by this exciting the helium nanodroplets to by using two pump energies, which is around 21 electron volt for exciting 1s 2s orbital, and then with 22.2 with 1s 2p. So in the plot, as you can see, in the y-axis, it is around it is about electron kinetic energy versus the delay speed. So the horizontal line over here, it represents the direct ionization of helium by absorption of one pump and one probe photon. And in the RHS of this, you can see the probe spectra, delay spectra. So in this case, you can see there are two main like peaks, which are D and A. It like for 21 electron volt for shorter delays, as you can see, only the peaks at 0.25 and 0.45, it is showing the broad peak which is dominating in this area. But in the case of a delay speed like 0.65, 0.85 and 2.55, it is showing a sharp peak and it is dominating. So it shows a time result photoelectron spectra. And further, can you go to the next slide? Yeah, further by fitting the spectra, Two further plots are like plotted, which provides information about the amplitude of these D and A peaks and position about that. So in the first plot, we can see it represents about the peak area versus delay time. And the second plot about which provides the information about the position, it provides information about like position corresponding to the various delay time. So as you can see, the peak D rises within the first 0.5 picosecond delay time, and then slowly decreases, and corresponding and accompanying the rapid increase of this peak A, which is represented here as in dashed lines. This kind of opposite trend is indicates the redistribution of population from D to A within this 0.5 to 2.5 picosecond. The energy of the peak, which is represented in the second plot, energy of peak D rapidly decreases within t less than 1 picosecond, followed by a slow decrease beyond 2.5 second. And the peak A slightly shifts from 0.9 to zero point 0.8 electron volt within time one less than 1 picosecond and remains constant. So the final peak position, which matches the electron energy expected for ionization of helium atom in the lowest excited singlet state, like for the 1s 2s orbital, it's around 20.6 eV, and the 4 eV is the constant, and for ionization potential it is around 24.6. So it provides around 0.8 electron volt, and this peak A is associated with according to this energy, it is associated either weakly bound to the droplet surface, or it is ejected into the vacuum, which is interpreted by the photoelectron spectra measured for various helium nanodroplet sizes, which I'm going to discuss in the coming slides. And it is based on the time-dependent density functional theory. So so this was for the singlet state atoms, and coming to the droplet size, for the droplets, the peak D associated with the helium located further inside the helium droplets, such that the energy it is energetically shifted up. So by considering the 21 electron volt and 4 eV is the constant, we are getting a similar output of something around 1.3 electron volt. And and the further things I'm going to discuss in the next slide. Can you go to the next one? Yeah. So now the question arises is that how can the extremely weakly bound ultra cold van der Waals helium clusters induce ultra fast energy relaxation by up to 1.6 electron volt within the 1 picosecond? So the answer to that lies in the potential curves of the helium excimer correlated to this 1s2p and 1s2s and 1s2p orbitals. So as you can see over here, as Joyce also mentioned earlier, so as you can see here is the blue shift absorption profiles is shown, and with respect to the atomic levels can be related to steep upwards bending of optically active A, D, and F state in the range of most probable interatomic distances. So this most probable interatomic distances is shown here as a gray circle, a gray dotted line circle. So in this region, it is the these optically active A, D, and F state are the most probable here. And when the exciting after exciting the helium in a nanodroplet, these diatomic states are like expected to be strongly coupled to form a band-like structure. So further relaxation by internal conversion by the crossing of potential curves of B and C at short interatomic distance is illustrated by the dotted arrows over here. As you can see, from this most probable region to also something around 20.7.5 like that. So this is the first. This is the step one of the relaxation, and further the local environment rearrangements occurs to accommodate the newly formed this 1s2p helium atoms. This is the step one of relaxation. So it is going to be further explained by using the time dependent density functional theory, by using the helium density distribution function. In this plot, you can see This is the helium density distribution. It is shown in terms of like the yellow dot, and this is the nano droplet containing almost around 1,000 helium atoms and having a radius of 2.2 nanometer. So uh, uh, so in this case, we can two cases were considered. At D is equal to 0, which signifies the helium atom would be at the surface of the nanodroplet, and in case of D is equal to 0.7 nanometer, it would be something around deeply inside the nanodroplet. Okay, so at D is equal to 0, the it locally compresses the surrounding region, forming a spherical dimple. And after that, after the after 0.4 picosecond, it slowly forms a after it is a spherical dimple, and after that it slowly flies off something around 0.8 picosecond. But in case of when the helium would be some deeply inside the nanodroplet, which would be around 0.7 nanometer. In this case, the formation of bubble is occurs around at 4 picosecond, and after that the bubble bursts around 4 picosecond, and then it flies off at around 9 picosecond. So this is nothing but this is just a correlation between what we have like I discussed earlier. Like, can you go to 2 slides before? Yeah, that again. Next one. Yeah, so this one, as I discussed earlier, so two types of the relaxation of helium nanodroplets occurs at two different stages. So with the first one, it is occurring from this band edges, and the other one is due to the local environment rearrangements that is occurring due to this formation of void-like structures, and which is explained on the basis of this time-dependent density functional theory. Go to the next slide, please. Yeah. So this is what it was to relate with correlate with the experimental and theoretical results. It is performed to observe what is happening, how the ultra fast relaxation is happening. It provides a complete picture of that. So can you go to next slide? Yeah. So coming to this plot, it represents the simulated time-dependent photoelectron spectra. It is the at the x-axis represents the delay time, and this is the simulated kinetic energy of these plots. What we observed in the previous slide, the droplets. So you can see very different at different position, like which is around 0.0 nanometer, 0.2 nanometer, like that. So these two is around the surface of the nanodroplet, which is around 0 and 0.2, and mentioned is like it's blue and green. So you can see, as you can see over here, the blue line, when the like helium atom is at the surface, it is relaxing quickly with some within something around 200 to 500 picosecond. Similarly, the green line, which is at 0.2 nanometer, it is also rapidly decreasing within this 500 picosecond. So other than that, the other like helium atom deep inside, which is deep inside the nanodroplet, it is the distance is around 0.4, 0.7, and 0.2. It initially first decreases till 0.9 eV, as it is mentioned over here. Then gradually it followed a slow decrement, and it like goes like that up to 2 picosecond. So the in case of the plot B, it represents a the theoretical average of what we have discussed in the plot A. Then this red line represents the droplet within the droplet, the peak T at 21 electron volt, and for the pK for the atomic singlet atom at 21 electron volt. This is this theoretical and experimental values are something are in good arrangement. So it provides yes, it is showing a good agreement between this theoretical and experiment results about this ultrafast dynamics. Okay, so the next slide. Okay, so this was all about that I understood in a very brief amount of time. So it was a very fundamental thing, and it is very curious. Like it is very like it is very interesting to observe few different things as well. So in conclusion, I would say like the relaxation dynamics that occurred in free nanodroplets study was possible in great detail using this ultrashort tunable XUV pulses. So it can be used for probing the photodynamics of more complex natural and synthetic nanosystems. As Joyce also mentioned that helium nanodroplets are very standard materials, and we can also use very different types of we can dope something with that, and then we can observe very different mechanisms or phenomenon with that. So in case of various regions of excitation of valence cell or even inner cells also, but the direct time result imaging of the bubble dynamics is at the present state very challenging, so given the small size of bubbles and the expected low contrast. So in terms of future perspective, the further development of ultra bright X-ray light sources is needed to enter the regime of atomic resolution in single cluster, and will then deliver a wealth of detailed information. So this was all about that our presentation. I hope it was okay, and Professor Mudris can like provide some more insight about that, whatever we have told, and where could have we could have some improvisations.
Yeah, thanks very much. I think you did very well given that this is, as you say, short time for preparation. I think you grasped all the important aspects of it. Yes, I probably am not the best person to ask questions, but is there any other one who has questions? Maybe from the Kassel team also, since we are interested in helium droplets for the future as well. Is it all clear to you, or do you have any questions, any remarks? Maybe just one question also to guide or to incite a little discussion about the like free electron laser and HHG scheme. You mentioned very correctly that this was tunable radiation. So it was, and yeah, I mean, you showed results for 21 eV and 21.8 eV. So obviously the wavelength or the photon energy was tuned and in the supplementary of the paper, actually, we show, do we actually, I mean, maybe we don't show, but there are more data where we tuned the photon energy to different values. Do you know how this can be done? How can you tune the free electron laser in photon energy? What needs to be done to change the photon energy in this HGHG scheme?
Do we have to change the undulator parameters of the radiator that will basically reduce the gap between them?
That's true. Of course, the radiator, so that's the main amplifier of the radiation, that like the gain profile given by this undulator needs to be adjusted to whatever photon energy we want to amplify. I mean, this is exactly actually the way how we pick a specific harmonic from the electron bunches that come from the modulator and then go through the dispersive section. I mean, you write very correctly that these like pre-bunched electron bunches support many different harmonics. So if we want only one specific harmonic to be amplified, then of course the radiator needs to be tuned to that specific harmonic to amplify it. But if we now, so you can probably guess that there's actually two ways of changing the photon energy. There's an easy way, and that is just by jumping from one harmonic to the next. Yeah, and that's exactly done the way you describe it, by just changing essentially the undulator gap of the radiator. So by tuning the radiator to the next harmonic that's present in the electron bunches, then we can just amplify the next harmonic. So this can be done really in a matter of like a minute or so. You can jump from harmonic to harmonic, but this is like jumping from harmonics. This means this is typically jumping in steps of more than 4 electron volts. So if we now want to tune from 21 eV to 21.8 eV or 22.2 eV, what you showed, that's much less than jumping from one harmonic to the next. So here we want to be able to like continuously tune the photon energy in a small range. How can this be done? Do you have an idea? I mean, yes, the radiator, of course, always needs to be adjusted, even if it's a small step in photon energy, let's say half an eV, still the radiator has to be adjusted, but that's not enough, because like the whole lasing process is not started in the radiator, but where is it started? So what else do you have to tune? So where does the HGHD process start in this scheme?
It's the seed pulse, so we have to tune the seed pulse.
Exactly. You have to tune the seed also, and accordingly adjust the radiator to the proper photon energy. And so how can you tune the seed? I mean, yes. So what kind of laser should the seed be then?
So you can have a broadband with a monochromator to select which wavelength to be used, because the output FTL pulse is highly dependent on what wavelength you are giving as the seed, right?
Yeah, but I mean, what's the wavelength of the seed?
It will be same as that, and we have to match it to the undulator resonance condition.
But no, it's not the wavelength that is amplified in the end. I mean, I think you said it correctly in your presentation that the radiator actually amplifies higher harmonics of the seed wavelength.
Yeah.
So what's the seed? It's written in the first line on the slide.
It's the third harmonic or from the OPA.
Yes, so it's the third harmonic of a TISAF. That's the same type of laser as you have in your lab.
Yes.
Just take the third harmonic, that gives you 4.8 eV or so. But the third harmonic of TISAF is not really tunable. So if you want tunable seed pulses,
OPA.
then then it's the OPA. And yeah, for those who are not so familiar, OPAs means optical parametric amplification. So that's a nonlinear process where you can essentially by frequency mixing two different pulses, you can choose the ratio at which the photon, like the sum of the, or is it the sum? No, it's the difference, right? You take the difference between two different photon energies, and that can the ratio between the two pulses created in the OPA can be changed, and by tilting the crystal and adjusting some components in the OPA, and in this way we can tune the photon energy. Yes, so we need a tunable femtosecond source to start the process with. So, but this is commonly implemented nowadays. Okay, Any more questions, any more remarks to this nice presentation? Okay, no, if this is not the case, then thank you very much. I think you both did very well. Very nice presentations. And that's it for today. We meet again on Wednesday. I'll give a little primer on coherent diffraction imaging, and we have another presentation on attosecond pulses. I'm sure this will be very interesting as well. So see you again on Wednesday. And
Thank
good
you.
night to India.
Thank you, folks.